So we are in the Scottish Centre for Macromolecular Imaging, which is a new world-leading centre for structural biology research based in the University of Glasgow. So the SCMI is a really exciting project. It brings together the combined structural biology resources of Scotland. So structural biologists from across Scotland have come together in a consortium to establish this centre and provide access to state-of-the-art technology for cryo-electron microscopy or cryogenic electron microscopy. The microscope we've bought for the SCMI is the first of its kind in Scotland and one of only a handful across the UK that are available to researchers to investigate the structural biology of important processes. So we're very excited to be at the centre of structural biology in Scotland and have that capability and be able to offer that capability to researchers from Scottish universities. So cryo-electron microscopy, we call it cryo-EM, is a way of preserving biological material at very low temperature and then imaging it in a very high-powered electron microscope. We can take the images of biological material from the electron microscope, process them in a computer and extract the three-dimensional structures of the molecules we're interested in. We can then, from those structures, build models at the atomic level to understand how those proteins or nucleic acids work. And we can apply these techniques to understanding a whole range of biological functions, from infectious disease processes to disease processes like cancer formation. And by understanding the molecules that mediate these changes, these biological changes that can lead to disease states, we can begin to try and understand how to interdict those processes and prevent disease happening.